saying this. What a privilege to be here. It's wonderful to join the family and colleagues who can help me understand more about Earl. Earl was gracious enough to have me in his laboratory in 1975, I think it was, where I was able to go into his lab and work on cats and do some really amazing things. He was gracious to let me in. I learned a lot from him. He was an amazing experimentalist. He was really fantastic at dissection. I learned that from him. I didn't spend a lot of time with him, but I really came to recognize his professionalism and that he was truly a kind person. So I was very grateful. Since Judy and Reggie are here, I have to say something about them. If you talk about standing on people's shoulders. So I grew up in Van Nuys, California. I was not supposed to go to school. I was supposed to go out and get a job. I was fortunate enough to be able to come to UCLA for $109 a quarter in 1971, and it was very important for me that I was able to work with Reggie and Judy in their laboratory, and I have to thank you for everything you did for me, because I would not be here without you. So they stood on Earl's shoulders, and I'm standing on theirs, so thank you very much. So you've seen a slide of this paper, this landmark paper from 1953. You've seen it 3 times now, I think, already. So here is Earl together with granite. You've heard about granite. Let me say just a word about Merton. Merton and Marsden had these famous papers on muscle wisdom, which was the idea that motor neurons could adjust their firing rates to accommodate what the changes that were taking place in skeletal muscle, and in that way kind of minimize the amount of fatigue in the system. So Earl certainly was trained by some major leaders in the field at the time. I was also amused in thinking about granite, because I hate reading his papers. His papers are about as dry and prescribed as you can possibly read. But all the information's there, and I've learned a lot from it. I don't know how much of that style Earl picked up. I don't know. You have to be the judge of that. In any case, so here's 1953, and people have mentioned Edmund Hillary a couple times. Nobody said anything about the Sherpa Norgay, but the 2 of them were the first people to summit Mount Everest in 1953. So there it was, and this year was also of interest to me, because that was the year I was born. So there we are with this landmark paper in 1953. The trace that you see here is a trace of the action potentials that are produced by these sensors that are out in the muscle when you stretch your muscle. These muscle spindle afferents fire in prescribed ways depending on how you stimulate them. And this was a figure that was taken from Earl's paper. You're going to see a couple of these sorts of traces from the work that I'm going to show you. And I'll move through it quickly. I do want to give you a sense of the legacy. So here we are. Here we go. Here we are with a diagram that I have for you. Here's a muscle, and out here we see this coiled structure, and this is a spring-like structure. It's a muscle spindle. I tell the students that if you wanted to design a sensor to be able to tell you where your arms, where you're positioned in space, I like to dramatize. I open up a pen and I find the spring and I put it right here and I say, there it is. Just put a spring in your muscle and have the spring be pulled or shortened or lengthened as you move that muscle. Put a protein in there that senses that change and converts the mechanical energy into electrical energy, and away you go. So since I'm at an engineering school, they seem to like that idea. And I have to say that there's some very sophisticated sensors that are being put into these exoskeletons that are basically following that format. Here's what I'll be talking about mainly today. Here's a muscle, here's the muscle spindle, and here's the sensor that's coming back into the central nervous system. As Reggie already showed you, these afferents, these sensory nerves, project widely throughout the central nervous system. I simply wanted to show you this because now you can get some sense of why proprioception and problems with proprioception is just so important. These are not just little sensors that go in the spinal cord and end there and just tell you whether or not you're being perturbed mechanically. They're actually going up to the brain. They're telling you exactly where your body is in space. I like to do with the students again. I like to say, well, how do you think you tie an apron behind your back? The only thing you're using to do that is proprioception, or touch the tips of your fingers behind your head. Not until you get to the target do you get any cutaneous input at all. Everything up to that point is from these sensors. Okay, so these are data taken from our laboratory to point out where these sensors project into the spinal cord. They're not only giving us some information about sensing where our body is in space, but they're coordinating our muscles when we do find ourselves in an environment where there's unexpected changes. We can walk across this room and step on this wire all of a sudden and receive feedback that coordinates our muscles, that allows us to keep going forward with the purpose that we had intended. Here we have a cross-section of the spinal cord. Each one of these dots is a synapse that's made by afferents that are color-coded here and distribute widely throughout this cross-section of the spinal cord. Okay, I want to show you some data from Tom Jessel's laboratory. This is Turgay K. These are his data from PNAS in 2014. And I'm going to show you how mice normally walk on a ladder rung test. So a ladder is a nice way to test proprioception because the animal's looking straight ahead, especially rodents, they're looking straight ahead. And what they have to know how to do is to place their feet in the right place on the ladder so that they can negotiate the path. So we're going to see here this mouse walk across the ladder. And of course this is slow motion. And we can see the markers on the various joints. This is a kinematic study. And we can see the animal does really pretty well with this. Now we're going to compare this performance with a mouse that has had its proprioception knocked out genetically. It's a mutant mouse where they knock out the muscle spindle formation and with this EGR3 mutation. And we'll see how they perform then. Okay. So in the knowledge that everything is okay with this mouse except for the muscle spindles, you can see just how important it is for this animal to have that input and just how devastating it is when it doesn't have it. So I want to tell you about two conditions where impairing 1A sensory neuron signaling has an important consequence. And these are studies from my laboratory that we've been performing over the last, I don't know, probably 2 decades. I'm going to start out by telling you our most recent studies with platinum-based chemotherapy. Now, it turns out that human subjects have a great disability long after chemotherapy has saved their lives, and they're disease-free, and they move forward with their lives. But for many years thereafter, they have great difficulties. They say, I can't really drive, and not the way I used to, and I feel a little clumsy, and I don't really have the balance that I used to have. There's another case, it's called chemo brain. Chemo brain is the case where they feel lethargic. They have a mental lethargy. So these chemotherapies can be very devastating for long-term quality of life. But as I say, they're very important for getting rid of the cancer. And we can see here that these platinum drugs that are used, oxaliplatin, cisplatin, carboplatin, they are actually the treatment added to surgery for 50% of all cancers. So these drugs are here to stay. We're hearing a lot more about immunological approaches, but these drugs are here to stay for decades. And so we really need to work out what kind of problems they're causing. Let's take a look. So here we have a rat. It's walking down the ladder. You'll see that the spaces are uneven. We're trying to make this as difficult for the rat as possible. And we change the spacing so the rat can't learn this task. We're trying to assess the extent to which this animal can place its feet accurately on the bars. Okay? Now the same rat was given a full dose of oxaliplatin adjusted and scaled as it would be to match to what the human would receive. And now we're going to allow that, we take them through the full treatment, and then we wait for 5 weeks. So this is 5 weeks after a full course of oxaliplatin, and here's how that animal walks. And you're going to see now why I showed you the video from McKay, because we see the same sorts of problem. So it looks as though there's some kind of, we would speculate, hypothesize that there's some sort of problem with proprioception. So what we did, we went into the laboratory and following the sorts of things that that Earl did in his lab, but working with rats instead of cats now. And instead of teasing fine filaments, which I did for many years of my career, we now just use micropipettes, and we actually penetrate the axons singly, and we get much better signal and noise, and away we go. Somebody came through my lab and asked if he could learn how to do this, and so we worked with him. And I saw him at the Society for Neuroscience, where he was presenting data that he had collected using this technique. And somebody said, is this hard to do? And the guy who I trained said, it's not hard at all. All you have to do is, if you were to fall asleep and your head hit the table, it would knock the electrode into the axon. So it's not quite that easy, but it is a very good technique. And what you can see here, in association with the animals that I just showed you, we can see here that normally, here is how the 1A afferent fires when we apply a ramp, hold, release stretch of the muscle. These dots indicate the instantaneous frequency, and here we can see that we get, during the ramp, we get a signal, a very high frequency, and importantly, during the hold phase, normally we get a sustained firing. I'm going to take just a second to mention Judy again. In one of her graduate program courses, we went through Peter Matthews' 1972 book on the mammalian muscle spindle, and one can find these sorts of figures in that textbook. You know, I was very sensitive to what Reggie was saying. We can conclude that either I'm standing on the shoulder of giants and continuing to try to push forward in the frontier with this sensor, or I'm a dinosaur and I'm just continuing to study the same things that other people have studied all along. I have to say that I'd be fine with being a dinosaur because I love these studies. It's so much fun to listen to the sensors in the audio amplifier. But I do hope I'm pushing forward a bit. Anyway, here we've got what one would normally see, and in the red, these are muscle spindles that are recorded from in rats that have had chemotherapy. So we can see that unlike what is currently in the literature, in contrast, what's currently in the literature is that the reason that we have trouble with movement after chemotherapy is that the nerves just die back, and they disconnect from the sensors. Some of them do, but that's not the only problem. In fact, the only ones that die back are in the feet and the hands. What we're showing here is that have problems throughout the body. A portion of the signal has been deleted. So what we might anthropomorphically suggest that what's going on here is that during the hold phase, this animal is receiving no information at all about what its position is in space. Once it achieves a position, it's not getting any information because it's not sustaining firing. And it's not only the 1A fibers that exhibit this behavior, but it's every fiber that we've looked at. Here are some other, here's some other ones. The group 2, also from the muscle spindle. The group 1Bs that are signaling muscle force. And these are the slowly adapting type 2 skin afferents that also, every one of these cells that normally sustains firing is showing an inability to do so after chemotherapy. 5 weeks after a full dose. We've carried on with some other studies. These rats can get pretty large when you leave them in the cage for a long time, but we've looked out to a year and they still have these sorts of problems. Okay, so what in the world is it? How are we going to figure out what is causing that problem? Well, the approach that we took was to say, can we take a normal animal and give it a drug that would reproduce the sorts of behavior that we see here? A drug whose actions we understand and know. A drug we know what it's doing. And that if we could find some similarity, then we might have some insight into what was causing the problem. So we started with a drug called riluzole, and then we moved on to a drug called phenytoin. Phenytoin you might recognize as Dilantin. It's used in epilepsy, And one of the things those patients complain about with chronic phenytoin treatment is ataxia, discoordination of movement. So we were able to reproduce what we saw with this chemotherapy agent by using these two drugs. And they gave us some real insight into what was going on. What they suggested to us was that we were blocking a persistent inward sodium current, a particular type of protein that allows ions to flow when the muscle is stretched, and that we were damaging that somehow. That's exactly what these two drugs do. They block that protein. Well, if that was the case, then we might know a way to fix it. So there was a drug that was available. It is available. It was used as a weight control drug. It's called lorcaserin or Belviq. It is a drug that increases persistent inward currents. And it was used for weight control because basically what it does is just jazzes up the nervous system. It just kind of heightens excitability of the nervous system, increases metabolic rate, and the idea was that would be a good weight, it would address problems for weight control. Turns out that as a drug for weight control, it's not very good at all. But so this may be another case that of the sort that you may know, the Viagra story. That was originally for controlling cardiovascular problems. Wasn't very good at that, so they recalled the drug, and the women gave all of theirs back, and none of the men would be willing to give their drug back. So or Rogaine. Maybe I should have used Rogaine. Rogaine's another one of those drugs that's been repurposed and very effective at another sort of problem. Perhaps lorcaserin will be good at assisting chemotherapy patients with regaining some of their normal function, because as we see here, it increases it restores the repetitive firing during static stretch. I have to say that I entered into the world of clinical trials and I had no idea how difficult it was. I do now know how hard it is to get patients and how hard it is to work with clinicians. And I've tried with Ohio State, and now I'm trying with Emory, and we're just having some real difficulty getting patients to subject themselves to trying lorcaserin to see if it'll be effective. We'll keep trying. Okay, so I'll end that portion of the talk by simply pointing out that we've now tried to move into the molecular realm to figure out what's going on. We remember that we had a hypothesis that a particular protein was involved, a particular ion channel. So we hypothesized, in fact, that it's the NAV 1.6 that has persistent current associated with it. We don't need to worry about the name, but the question was, does it even exist in the muscle spindle? And here maybe I need to be speaking to Earl about this, because it's turned out that I was so surprised to learn that after all these years, we don't understand the molecular mechanism by which these sensors are signaling. We really don't. 3 years ago, there was a protein called a piezo-2 that seems to be responsible. It's a protein that when it's mechanically disturbed, it allows current to flow. That was the first time that we had some indication of what the mechanotransducer was in this sensor. Turns out that we don't even know what the encoders are for these firing properties. And so in this particular study, we were trying to identify whether, for the first time, whether or not these channels were present in the muscle spindle. And my colleague, Dario Carrasco, is fantastic with immunohistochemistry. So here we see that we were on the cover of the Rolling Stone and had our work highlighted in this particular issue, where we see this muscle spindle, these coils. The green is simply a structural protein to let you know, let you see the structure of the coil. And here's this little red bracelet right there that is in the encoding region and may be the culprit for causing the problems that we saw. We haven't gotten to that point yet. Okay, I have just a couple more slides to show you. Here's the second thing that I wanted to tell you about with regard to what problems might occur with these spindles. And in this case, what we're going to consider is what we see normally here. Here's a muscle spindle. These afferents project into the spinal cord. where they make a monosynaptic, they directly contact motor neurons. And that's the basis for the tendon jerk reflex, right? So you activate these spindles, they go into the spinal cord, they directly excite motor neurons, and you get a muscle contraction. That's exactly what this is. And it turns out, and my PhD advisor was prominent in making these determinations, that each single one of these, every single one of these 1A afferents contacts every single motor neuron that supplies that muscle, without exception, every single one of them. And when we apply stretch to a muscle, normally we get this signal of excitation that's transmitted to each one of these motor neurons by each single 1A. Now it's important in that functional anatomy, because what we found after we cut this muscle, or cut this nerve, sutured it back together, allowed the nerve to grow back into the muscle, we ask ourselves, okay, does this system function normally? There must be some problem, because just like the chemotherapy patients, long after a nerve has been severed, sutured back together, grows back into the muscle, these subjects still have problems. And as the mantra in our laboratory has been that recovery is not synonymous with regeneration, that regeneration alone is not going to give you recovery. Why? What was going on? Well, we started out by studying how these afferents were encoding information, and they looked normal. That we couldn't see any change. But we looked inside the central nervous system, and what we found was now a lot of the excitation was much smaller when it existed, and in about 40% of the cases there was nothing there at all. That was absolutely diagnostic, that some of the branches of these cells inside the central nervous system were pulling away. They were pulling their synapses away from the cells. So I was working with my colleague Paco Alvarez, who's an anatomist, and anatomists don't ever believe physiology. Paco would say that he had to see it before he believed it. So Paco got us, he joined the project, and he looked at alpha motor neurons. This is a motor neuron, and this is a motor neuron. Normally, we see that these motor neurons are studded with a high density of these red dots. These are the synapses, centrally, that are made by 1A afferents. And this is what you see normally. But after regeneration, this is what you see. Those afferents don't grow back in. In fact, we've actually found, and I won't show you the data in the interest of time, but we've actually found that the axons that come into lamina 9 to the motor neurons, they lose their synapses and they pull away. This is not just simply synaptic stripping, for those of you who know that. It's just, it is a virtual retraction of that branch of the neuron. And even worse yet, I guess I'll show you this. This is the case where we look at the percent depletion, just how many of these are lost. We look at the percent depletion under two different conditions, where we cut and suture and allow regeneration, or we cut and tie off the nerve so you get no regeneration. And you can see virtually no difference long after the injury. That nerve's going back into the muscle, and it's just not restoring the central connections. So normally, this is what we see, and I've showed you this slide before. I'm going to represent the areas within the spinal cord by these spheres. So there's kind of a dense area of synapses here, and that's represented here. It's kind of light in lamina 7, where we expect to see all the 1A inhibitory interneurons. I was surprised not to see more. There's not much there. And then down here's where the motor neurons are. So I'm trying to represent how the density with these spheres reflects what we actually see with the data. After regeneration, we see the following. Number one, we see that all the motor neurons, that many of the motor neurons lose their synaptic connection. And this branch here, it actually retracts back. These synapses, they get smaller in size. Look what happens back here. The number of synapses here doubles in number. So I particularly wanted to show this to my colleagues who've been studying spinal cord transection all these years, because I'm thinking to myself that this is kind of a model of what might be expected after regeneration of descending cortical motor neuronal cells, for example. That just because they regenerate does not mean that all of the synapses that are unaffected by that injury are going to just fall in line. We'll get regeneration and everything will be fine. I think intuitively we all knew that that was probably too much to hope for, and I think this is good evidence that that there's a lot of reorganization that we're going to have to deal with. Okay, then, so I showed you problems. What are the problems with proprioceptors? I showed you two, where we have problems with signaling out here, and we have problems with transmission in here. And with that, then, I wanted to mention my colleagues at UCLA, and here they are. Again, many thanks to Earl for allowing me to come into his laboratory and do my first studies. I was there with Barry Boderman when he was a PhD student, and I think I was kind of like the little brother that I think aggravated you on several occasions. Yeah. And then, so at Emory, I've had quite a few colleagues, also at Wright State when I was there, and now my colleagues here at Georgia Tech, and have managed to maintain some support from NIH. So with that, I just want to thank you for the time and the privilege to be here.